Good morning, friends. Myself, Dr. Naval Singh Todawat, and we are having this video series for the BSc TY student. Whereas we are focusing on the genetics and genetics engineering. So, in that, we saw regarding the genetic engineering that how the genes are manipulated. Well, a target and the desired genes of a specific aspect of a specific character is isolated from the organisms and which is reintroduced for that a particular desired character. While genetics or the genetic engineering is the deletion and addition or duplications of the desired genes in a specific way. So this is the process in that we replace, add and delete the genetic unit from a whole chromosomes. So in that we saw there are 6 steps process which are followed in the case of the insertions of the desired genes. Mostly we see the bacterial cells. Plasmid is used as a vector. Plasmids are the circular DNA which just lie in the bacterium cell, and the desired genes from the cell or the plant cells are added to this plasmid. Initially, the plasmids are cut with the help of the enzymes, and the desired genes are attached to this with the one ends at the target sites, and these plasmids, what we can say again, recombine with the targeted genes which is reintroduced. Well, now what happens, we see that what the transfer of this targeted genes or DNA is done by the process of the host. Well, the recombinant DNA is transferred in bacteria. Well, it is again transferred to its host one, that is to the bacteria one, with the help of the different equipments, or we can say with the help of the same things are cultured, same species of bacteria are cultured and replicated. The bacteria recombinant DNA is cultured in the medium, nutrient medium. So the isolation of a gene cloning the host cell containing foreign DNA is isolated and allowed to multiply, forming a clone having the millions of identical copies of these cells. Such a process is carried out in a medium. Sometimes we use the nutrient agar or Na, or sometimes we also use the nutrient broth to achieve a large amount of a target gene. Whereas the insertion of the target genes in the recipient organism means uh, what the target or the specific character genes we have selected, now we are going to introduce in that particular recipient organisms, in that particular organism in which we desire to have the changes in such type of organisms, in such type of plants, or you may call it animals. Now we'll insert the genes, or we'll insert the recombinant DNA in the recipient organisms. While the target gene in produced during genetic engineering are isolated by different types of ways, which are those the target gene can be transferred into organism that requires it to get the maximum benefits. Well, when we insert a specific character genes into a recipient organism, naturally we wish some the best qualities or the best characters among those organism we wish to develop the characters in that particular organism, and that is why we try to insert, or we practically insert the gene which we introduce to that the recipient organisms. The target gene is a plant is transferred by bacterium, that is the Agrobacterium tumefaciens. These are the Agrobacterium tumefaciens which develop the tubers or not like a structure in the roots of the plants, and we all are aware of this Agrobacterium tumefaciens or micro injections. This is the gene transfer vector. So I repeat the statement, that is the target gene in plant is transferred by the bacterium, and most specifically, or the most what we can say that the common specific bacteria, that is the Agrobacterium tumefaciens. This genus or species of the bacterium is well known for the productions of the tumors on the roots of the plants, and this bacterium helps to inject the gene transfer vector. Application of a genetic engineer. There are different types of application of genetic engineering. Nowadays we are aware of the lot of the plants which are genetically modified in the our fields. They are also to be called as the GMO, that is the genetically modified organisms. It is a long form of that GMO, and these organisms, such as in our day-to-day life, we have seen the rice varieties such as the a brown one, the long one, and the short one, and again there are different types of the varieties which shows the morphologically structured and somewhat the what we can say that the genetically modified characters such as the smells and the taste of that rice of the grains are also modified, and they are chosen by the specific character as we seen as we study. The genetically modified organisms, that is the GMO, is also called to be transgenic organism. They carry foreign genes. They carry foreign genes or foreign DNA. Such organisms are of two types: genetic modified organisms. One is transgenic plant. As we spoke, in plant we introduce the transgenic vector into the Agrobacterium tumefaciens, which is associated with the root nodes of the plants. So while transgenic plants are genetically modified plants produced by genetic engineering, such plants have more resistance power against stress and disease. This is one of the best desired character what we take from this genetic engineering. The nitrogen fixing quality can be transferred to plants by inserting NIF genes from bacteria to plants, that is the nitrogen fixing bacteria. Then thus plant carry the NIF genes for the nitrogen fixation. For example, tomato slow ripening, golden rice vitamin A, potato high protein content. All these characters are increased in a specific species due to the transgenic plant, or due to the insertion of the targeted genes in that specific species. The genetically modified animals. Now, this is regarding the plants, and as we can do the genetic modification in organisms such as plant as well as the animals. What about the animals? In genetically modified animals, which are transgenic animals, such as bear more protein content in milk, improved meat quality, etc. Which are those? So we'll have a little overlook on the genetically modified organisms. As we spoke about the plants, we saw the tomato, potato. Then we also look out in the common market, that is the cotton. Then brinjal. There are lots of varieties which are genetically modified in plants for a specific character. As I want to give an example of a tomato or a brinjal, in most of our region, according to the phytogeographical region, the peoples consumes different types of a taste of a brinjal or a different likes different types of shapes of a brinjal. In some region, we see that the tomato, some people prefer of a big and large size, whereas in a few regions, such as in the what we can say in the westerns, the people loves to have a tomatoes which are of a very small size, seems to be like a grapes. And on that basis, according to the consumptions of the peoples, according to the market demand, such plants or this such a fruits crops are the genetically modified on accordance of that specific characters, and such a desired genes are to be transferred into the plant with the help of this bacteria. And thus this whole process is to be known as the what we can say the genetically modified organisms or the genetically modified plants or the genetically modified animals. Well, now we are going to have a look on the animals, or how this is also used in the medicines. It also used in the gene library and different field. Okay, look out at the medicines. Different kinds of antibiotic, amino acids, interferon, vitamins, hormones, enzymes can be produced in large quantity in the short period of time, whereas gene therapy can be done with the help of the genetic engineering. Well, we all are aware of the insulin. Nowadays, in the patients of the sugars, insulin is playing a main role in the sugar patient. What it does, the insulin helps to maintain the sugar level in the body of that human. Whereas it is a demand in the market to provide the insulin from the artificial insulin, whereas our body itself produce the insulin. But when this structure or the physiology of our body disturb, then at that time it is recommended to inject the insulin from the artificial way. Gene library, the collection of a duplicate copies of a fragments of a DNA, is known as a gene library. Well, we all are aware of the book library, seeds library, of the blood bank library, and so on. Now, in next advanced stage, we can also assume or we can also collect the genes of a desire, of our wish of that characters. We can collect and preserve such type of genes. The collection of a duplicate copies of a fragments of a DNA. Okay, when the fragments of a DNA, when we cut the DNA with the help of the enzymes for our desired character, these cutted pieces of a DNA, this fragmented DNA, can be collected in a gene library, whereas which can be produced by genetic engineering. Such library can be used as a reference for the researchers for the future research. So I repeat, the collection of a duplicate copies of a fragment can of a DNA is known as gene library, which can be produced by genetic engineering, and such gene library is be useful for the researcher in the futures. DNA fingerprinting. We all are aware of the DNA fingerprinting. Fingerprinting is mostly used in the friends. Well, in a DNA fingerprinting is a method which is used to identify an individual from a sample of a DNA by looking at a unique patterns of the DNA sequences in the DNA. Well, I repeat, DNA fingerprinting is a method used to identify an individual, an organism, from a sample of a DNA by looking at a unique patterns in their DNA. This technique is also based on a genetic engineering which helps on finding the relationship among the organisms. Whereas in a society, or most of the time we have heard about the cases or about the matters which are run and how the kids are identified of the parents, or how the parents relations are shown with the help of this DNA fingerprinting. Most of the times in the DNA fingerprinting is also used in the criminals identifications, and in the any matter of a what we can concerns. If a concern matter of a DNA fingerprinting, we generally have heard, or we also saw in the society in the investigations of a departments. So, friends, this is one of the technique which is developed from the genetic engineering. As we told that offspring, the relationship of the offsprings and the parents are been identified on the basis of the DNA sequences or the DNA's pattern. Okay, look out at these diagrams. There are three columns. One is the left one, and there is a horizontal, sorry, vertical line among them, and you can see the suspect 1 and suspect 2. Illustration showing a comparison of a DNA fingerprint from a crime scene and a DNA fingerprints from two suspects. The DNA fingerprint from suspect 2 matches that taken from the crime scene. Image credit Genome Research Limited. So this is one of the best method to identify these suspects. This is the crime scenes. Okay, suspects 1 and suspect 2. Okay, look out at the pattern of the DNA, which one is matching with the crime scene. So obviously the suspect 2 is matching with the crime scene DNA patterns. Look out at this pattern, we can easily identify. See the pattern of the DNA. There are two sequences which are very on a equal positions, keeping with the equal distance. These are the two. Then there are again two, this one with showing the different types of variations in the distance, and the last one is one. So look out at this one, completely different, doesn't match in any source or in any condition with this one. So this one, that is suspect 2, is matching with the crime scene. Okay, this one is the environmental pollution control, how one can control the environmental pollutions with the help of the genetic engineering, with the help of the gene which helps in producing enzymes that break down the petroleum products into less harmful compound can be transferred into bacillus. Such a genetically modified organism helps in reducing the pollution. Uh, Whereas nowadays, I think so that in the last year, there was an invention of the bacteria which consume the plastics. Uh, this research have been supported by the Department of Botany, Pune University. In that, the bacteria used to consume the plastics and the polymer present in that plastic. What ultimate, the plastic doesn't dissolve in the atmosphere as like the organic matter, such as the leaves and dates and decades. And these plastics ultimately increases the pollutions in the environment on the earth. And if we introduce such type of bacteria more and more that consumes the plastic matter of this earth, ultimately it will help us to decrease the pollutions of our this green environment. Production of vaccine and antibiotics, this is, we all are aware of these techniques. It can also be utilized in the production of vaccines as well as the antibiotics. Development of resistance of a crop varieties, lots of varieties in the uh, markets. We see that with the bt, what is the bt, bacillus thuringiensis, these varieties are very common in the market nowadays. And these varieties bacillus thuringiensis, what it helps to regulate, to check or to resist the diseases and the pest of that particular crops. So genes are isolated that can be inserted on crop plants to increase the resistivity of that particular plant. Bt genes are isolated from Bacillus thuringiensis, which is used to increase the resistivity of a rice as well as the maize. So we all are aware of this rice, maize, bollworm, BT, cotton resistant varieties, all are aware in the markets, and from a decades these varieties are serving in our farmers' fields. Well, nitrogen fixation in non-leguminous plants. Well, we all are aware of that the leguminous plants, among all the what you can say cotyledons, mono and dicotyledons, among all the families, leguminous plants have the more capacity to fix the nitrogen bacteria from the soil and from the atmosphere, and which gives to the plants for its survival. Well, where in the non-leguminous plant we can insert or isolate the NIF genes, which is related with the nitrogen fixation bacteria, and transfer to the non-leguminous plants to increase the soil fertility as well as the crop productivity. As the soil fertility increases, ultimately the plants gets all types of the nutrients from the soil, such as the macro as well as the micronutrients, whereas the nitrogen, potassium, phosphorus, calcium, magnesium, and so on. Whereas the micronutrients, what we can say, iron, zinc, boron, and all that 16 or 17 types of the macro as well as the micronutrients are required for the plant development and should be present in the soil. But there is the variations in the soil patterns, in that condition we can use such type of the techniques to increase the fertility of our soil and ultimately to increase the productions of our crop. Friends, I think we should stop today's here. If you have any questions and doubts regarding all this topic, you may contact me on my cell phone, and you also may message me on my WhatsApp number. Till then, thank you, and please try to revise it and focus on this all the terms and terms used in this genetic engineering. Thank you and all.